Thank you very much for the organizers for this opportunity of talking in the synthetic biology community. For me, it's an honor just to be here. And then thank you for your interest in this session and opposed to competing ones. Okay, I'm going to talk about automatic design methods. So here the idea is the following. We are tired to work, so then we want to make the computers work for us. Okay, so we would like that the computers do the job, they do the design. It gives a nice DNA sequence, and then we synthesize with a robot, of course, and then the robot will do all the cloning, and we'll have everything. Okay, so then that is a long-term goal. So, okay. That's in this way I don't forget to thank my team. And then they are all here. So, and then they are in strategic places in the audience, so just to control you, just in case. Okay, so we are going to, how we are going to do this unsupervised computer design methods. Okay? So that's the whole idea here in this talk. Okay? And the thing is not, I'm not trying just to of course, replace the humans yet. But here the goal is the following. There are many, it's a huge complexity in biological systems. So it would be nice that the computer help us to design. So we have been in some sessions, if you were fortunate enough to go to the talk of Tania Corteme, you could see the things you can do with automatic design methods in the design of proteins, for instance. Okay, very good. Okay, so in the outline of talk, I'm just going to introduce you to the methodology. Here, we aim to evolve as genomes. But we are going to restrict that. We are going to make very crude approximations, okay? But that is the first step. So we want to, that with the time, improve this methodology. Or the community will contribute to improve the methodology. Then we'll just design, we'll show you the design of heterologous devices, just made of transcription factor and enzymes. And then I'll show you how we will model the cellular chassis, and then I end up with some conclusions. Okay, so then in the in silico genome evolution, so that's we call it in silico evolution, in the sense that we simulate a replication selection process, but in silico. But as we do it with the objective of design, so optimizing. So that's a clear difference with nature. In nature, evolution has some selective pressures that sometimes changes there, but here this is optimization. So we don't want to reproduce nature. We want to make an alternative nature. Okay, so then we would like to design the automatic, to make automatic methods to design genomes with a targeted function. And the problem here is that the mapping between protein and DNA is difficult. So going from DNA to protein, okay, it's just do some similarity searches, and so it's pretty easy. But the opposite, the mapping from protein to DNA is hard. It's very hard. So how are we going to do succeed with this task of designing new genomes? Because here what we want to design is DNA. Okay, so then here it comes that, as I'm going to show you, the idea that, okay, for some systems we know the mapping between protein and DNA. What I mean by that? I mean the following. For some systems we know some proteins that have some given functionality that can be worked that they can work with those functionalities in different contexts, and then we know the sequence of that protein. So, and I say proteins, but I can say regulatory regions. I can say other types of things. So these are and it came out to be the biological parts. Then, and that's very interesting, because then we are going to use that, okay, and then we are going to extend our methodology. So I'm going to show you to incorporate those biological parts. They will be biological part models, because they are just computational models, and to assemble them in the computer and to explore the richness of in the genome space. Okay, and so that's our way to circumvent this the problem of not having this protein DNA mapping. Okay, so in order to go to achieve these goals, we have been little by little developing different softwares for different aspects in the genome design. Okay, so then I'm going to talk to you about aspects about only transcriptional and design of transcriptional circuits, metabolic circuits, or how you combine them. So if here you can just see a sketch of the cell. What is here? I have only marked some modules that are relevant for this talk. So, there are all these input-output. By that I mean also some signal transduction. As I mean that the molecules here and here, they are not necessarily the same. And then you have the cell machinery. We have the regulation machinery, and then the metabolism. And then here you have here is like it's a module that represents a heterologous system you put in the systems in the bacteria, for instance, that itself has all those elements. Okay, so then you can just express, I don't know, a pore in a protein, or you can just do whatever heterologous thing. Okay, so now what we want to do is to have to start from a random genome. So that's our promoters and their coding sequences that are polycistonically regulated by that promoter. So here I have noted in gray transcription factors and in green enzymes. So then we are just making a difference between both of them. Transcription factors just carry a genetic load, so expressing them will cost resources to the machinery. And expressing enzyme, if you do it in an intelligent way, it will benefit the cell, because you may metabolize something that you could not metabolize before, and then you could use that to grow better. Okay. So we can just at the beginning go and do simply an analysis of how we evolve genomes only using transcription factors. Okay, that is we published that in our GeneT 2.0 software. That is freely available it's open source. Then we'll move to the metabolic space, where just we have only enzymes. Okay. Then a preliminary version that you can use that for the design, de novo design of metabolic pathways for biodegradation or for bioremediation, so or for bioproduction, and was published recently in bioinformatics and in collaboration with Kristalla Prader at MIT. That very nicely and publicized also this talk here. Okay, and then there are those approaches. They don't use these standard biological parts models. So we thought that it was the way to go to incorporate, as I said before, the mapping between protein and DNA. Okay, so then that we created a software that's called SM Parts. Also, it was published last year. And then this software is just assembled in silico, and it's just standard biological parts. And then our new software, GeneTDES, is using that inside. Okay. Then we can also use a chassis model to estimate cell growth. So that's here where it comes in the modeling of the system. So here we just, as a fitness functions, we can use among one of the fitness functions the cell growth. Okay, so then now I'm not going just to go into all details because here the goal is just of this talk is to become you aware of the tools we have, and then how far you can go with this modeling, at least in our team. Okay, so then let's go, and then let's see how we do this, the design of genetic devices and in heterologous systems. So that's the idea is just we use Monte Carlo. So what we do is we just start from a random from a random initial circuit, okay, and then we just make what here is called mutation. So we make a change, a local change. It can be a change in parameters, can be in kinetic parameters, or change in the topology. Or you can replace one promoter by another. By that, you are changing the topology, or you may be only changing the parameters. And okay, so then, as I said before, we could only just restrict ourselves in a transcriptional space. So here, then, the method is easier. So we just use very simple nonlinear models we saw in the previous talk, and you were just using it. And then with some ranges, okay? And then we just also use some parameters and then some changes in either in the topology or changes in the parameter according to the set of probabilities. Then our circuits that are, we start from a random conformation, and then we just proceed to evolve that to something that is, we have a better fitness, and then now I'm going to tell you what is the fitness. And the circuit may have promoters that have a combinatorial logic. For instance, we saw before that the promoters in the previous talk that had some non-trivial combinatorial regulation. And then as a score here, we have used, or we have selected a gene or some genes as reporters. So for instance, you may imagine that among the genes you are interested, you put polycystornically a GFP, so then you monitor the concentration of GFP. Okay? Then in this score, what it does is U in Z and in black is what you would like to have, the behavior. And then in Y in red is the actual behavior of the circuit. And so then the score is just the area between both curves. Okay? So it's the simplest score possible. You want that the system has a dynamic behavior given by a function. Then you just simulate a circuit, you compute its dynamics, you see how close it is to the desired dynamics. It is close, good. You accept that circuit. It is not good, then you go to the previous circuit you had before doing this modification. Pretty simple. Okay, so then these are the sequences we have designed. We have designed a circuit that have, for instance, some, they work as logic gates. Okay, so then, for instance, okay, so I don't want just to get off a screen. And for instance, here you see that you have U1 and U2 inputs, and then when you those implement in this time behavior, you can see they implement this truth table for a gate where only you have 1 in the output when you have 1, 1 in the input. So this 1 means that the concentration is higher than a given threshold, and then you see that only you have to have 1, 1, then you only get 1, the dashed line that is the NZ, the expected behavior. Okay, so almost it behaves as it should be. Okay, so this is the score is quite good. Then when you implement this, then you have to go and then to molecular details, then to design the parts. We have some experience of some years of doing protein design, and then this is Maria Suarez that has done this work. She's in the audience. Okay, and then but that is another talk. Okay, so we designed logic gates. I said and showed you the AND gate, but we have other examples. Then and we also decided some fancy memory devices that are more stable than the usual switches people use. So these are computer scientists. They know more about JK latches. And my students, Guillermo and Javier, here in the audience. Myself had to learn about those engineering things. Then also we design oscillators. And then all those you can go to the literature in our website. All the PDFs are there. Okay, so then you have here examples of all the devices, but I'm not going. So then this is an example of showing you that our JK latch exactly behaves as it should be. This is the truth table of the JK latch. And it's not the point here just to go to the analysis, but if somebody in the questions wants or outside, we can just talk. Then, it's very important for us to go forward and just to use models for biological parts. So then, and here we have made models that for promoter ribosome binding sites, coding sequences, and terminators, and then these are shown here is pretty simple models. But the nice thing is just we can assemble them and then construct many of the possible transcriptional circuits you can find in the registry, for instance. But so then you can see in the supplementary material how to construct. We put some examples, and then you just do it very quickly, just a few seconds. And here the nice point is that if the community engages in characterizing biological parts and putting that in SBML, we use SBML as a standard system biology, then software like ours and other softwares, we are not the only one. After in our publication, there have been other publications with similar things or more sophisticated things. And then people can just do design very quickly. So you don't have to, because designing all the differential equations by hand is very time consuming. And you have to explore, you want to explore many possible refactorization, let's say with the toll plasmid in the previous talk. You want to test all those possible refactorizations, you become mad. Okay, so then that's, from that you can just assemble whatever circuit is in this form. Okay. Then you can also go and do moves, but only in an enzyme. Okay, so then here, our idea is just to make smart moves. So come on, there is no point just to put in a circuit an enzyme that is completely uncoupled from the metabolism. By completely uncoupled from the metabolism, I mean that it's either its product or substrate is not doesn't have a precursor in the metabolism. Okay, so we use a retrobiosynthetic approach to find from a given compound pathways into the chassis, to the metabolism. Okay, so that can be used as a design tool, and this is how well we saw it in our publication. Okay, but you can also use it here as a tool for exploring the metabolic enzyme space or genomics. Okay, then also we have other moves for the enzymes. You can just change from operon. But here, the important thing is that when we put an enzyme, we put the whole metabolic pathway. Okay. Then, so that is the Sharkey, as I said. So the idea is simple, and let's assume that you would like to ask the following question: I would like to make E. coli to produce glucaric acid. Okay, and then you want the computer to do this job because you have better things to do. So our software it gives you automatically the answer. So there are some here. Some enzymes that they were not in the database, so these are more trickier. But so it gives you a pathway, but that we use Keck database, and then it is using different organisms. So of course, later you will have to use codon optimization for your organism of choice. In here, all the work was done in for E. coli. So then the idea is just simple. We have if this is our glucaric acid, and then these are the you have here in circles and metabolites, and in lines you have enzymes. Then you just can just go back trace until you just arrive to a host compound. So we have an algorithm to do that, okay? But it's simple. It's a heuristic algorithm, okay? And then we design things like, for instance, the previous glucaric acid pathway. But for instance, you can also design degradation pathways. So in this case, is toluene degradation. So we saw previously in the previous talk that there are also some degradation of poisonous compounds. And then here you see it just produces all the pathway. And then until you are plugged into the metabolism. Okay? So then it gives you all this the set of enzymes and their genes. Okay? And then, of course, it's important just to score that. So it's just there are so many pathways. So then you use some score that is based on cell growth. But I'll explain a little bit later. But there are other interesting things. So here is when you design an organism, is the design of transcription regulation. Not the design, the description of the transcription regulation by the chassis. Okay. Then here we use microarray data, and then from microarray data we have developed a methodology that infers the global transcriptional network. But this, so then, in fact, also we have developed a methodology that generates some from artificial genomes, as I showed you before. It generates synthetic microarray data. And it's important that because then we just plug this, as I'm going to show you a few slides later, couple that with the metabolism. So in our transcriptional and global network is able to reproduce some transcriptional data, and as you see here in gray and in black, from some macroarray data. Okay. Then we need also to describe in cell machinery the resources you are taking from the cell machinery. So here it's a collaboration with Drew Endy at MIT, and here is the idea is pretty simple. Is we from the 80s has been characterized what is the sequestration of the quantity of ribosomes that you have in a cell, and how they are sequestered, and when you just have, for instance, you have higher growth. So, for instance, here, if you have for a given growth, you know the quantity of ribosomes or of polymerases. So I call it cell resource as a collective name for that. Okay. So then, if the system, of course, it grows more, then you just are taking more resources. So then, here the idea is just you can use that and then just assume that the only cell resources all free available ribosomes that you are taken, and then you just can compute if the system you are consuming ribosomes, for instance, then the system will have a smaller growth. Then just you go here and then you can compute an estimation of what is the effect on growth due to have less ribosomes. Okay, so then we just here we validated with some growth media LB and supplemented M9. And then here then the whole point of this is just to couple everything. So we would like to just compute use growth, and then we have, remember, we want to have evolved genomes where you have only transcription factors and enzymes. We have simplified. We don't consider for the moment RNAs and all that. Then transcription factors have a genetic load. For the cell, it's hard just to have a protein that the only thing that does is binds to other to promoters, and it doesn't contribute to the real thing, to growth. Okay, But sometimes it's useful because what happens is that in a system, it may shut down a whole pathway that is not needed because you cannot utilize a metabolite. Okay, so that's the nice thing, that there is a trade-off between this cost and benefit of the enzymes. So we use a metabolic model. It's very simple, a decoupling between the time and behavior in transcription from metabolism, and then assuming this decoupling, that the metabolism is much faster than transcription, then we use flux balance, and then we use some feedback loop in order just to compute self-consistently the growth. Okay, so just to finalize, let me just mention that we have another collaboration with Cristala Prader, that is in the computational protein design of some enzymes that are missing in some metabolic pathway. Okay, this is a shorter pathway than the one I showed you before. It's a shorter, but then the enzyme doesn't exist, so then you have to do a computational protein design to do that. And then here also is our consortium that for hydrogen production that Phil Wright was speaking, and then. Okay. So this is the software that we have just related to this talk. Okay? And then as a conclusion, it's just I have presented some automatic procedures to evolve in genomes, simple genomes, I have to say. But in these procedures, you can use a fitness, not only time behavior, but you can also use growth. Our growth combines cost and benefit. We still, this is ongoing work. We don't have results, so I didn't show you results with the circuits. You can just grow with that. Okay, we are working on that. And then we have I show you results with toggle switches, oscillators, etc. And then let me just acknowledge my people, in particular, Guillermo Rodrigo and Javier Carrera, who carried out the work in this talk. And then you can go to our website, syntheticbiology.org, or synthbio.org. And I also thank our collaborators, but in particular only, Drew Endy and Cristala, I put it, that are the ones relevant here. And also, in this talk is the ones relevant here. And then this, so then so you can spot the people who just did the work, Javier Carrera and Guillermo Rodrigo. And then the other people, and then just the funding agencies, and also just to you for your attention. Thank you very much.
Thank you very much, Alfonso. The paper is open for discussion. Back there, please. Thanks.
Okay, no, I think you can hear me out there. Basically, what I was wondering is, you presented a situation. Okay, thanks. Okay, you presented a situation where you were talking about specifying to your software, creating glutamic acid, and basically it finds the path for you. Now, how exactly is that done? I mean, how do you interact with this software?
Okay, you interact with the software just by just giving which type of a metabolite you.
Right, but I mean, how do I do it? Do I say, do I type in glutamic acid? Do I type in, the smiles of it? How do I make that kind of request? How do I interact with that system?
Okay, so you can just we have a list of you can just go to the list. We have several possibilities, but one possibility is just go to the metabolite, because there's we use a cake nomenclature, and then in the cake nomenclature, that was a nice thing. You have only one name, but for instance, Brenda is a more complete. At least for some pathways we were looking, it has a lot of synonyms, and then it's terrible.
Yeah, please.
Okay. So your system is really good, and I think it could save people a lot of time. when they're designing their systems. But you said that you assume that you can change these rates, and you assume this promoter library. So have you thought about constraining the set of things that you could use to things that we have good measurements for, so that people could use your system right now to design something without having to wait for all those things to be measured?
Yeah. Okay. No, yeah. That we would like. So for the moment, when you do automatic design, you constraining yourself is always not a good thing, because we do you restrict your search space. So here what we do is there are two things. Parts when they are characterized, and they have some or they will have error bars. So we are anticipating a little bit into the future, because the number of parts that are characterized is very small. Okay? So then another important thing is that we have the concept of evolvability. The concept of evolvability means that, and I think that in this talk of Tania Corteme, and she mentioned, she called it in a different way, that is, you can have a given promoter or a given enzyme or whatever, and in your fridge, so you have the plasmid for that, etc., and then you know how to mutate that, for instance, to for the operator side of a promoter, just to mutate, it's like to spoil the binding of the transcription factor to the operator. To improve that is too hard, okay? But to spoil that, okay, and then increase the kinetic constant, that's is a different matter. Come on, it's not difficult, okay? And so it's much easier to spoil things. And then with enzyme, you just do alanine mutations in the active site or near the active site to decrease the activity. Okay? So then these are evolvable. So then, but the thing is, you can have in the fridge so many things that you don't need to have all possible things in the fridge, and then you just put it in the software, because you know your fridge is not big enough. Okay, so that's our idea. Then in one day, and even if they are very well characterized with some error bars, the error bars corresponding to evolvability will be even larger. Okay, so then still there will be some parts that doesn't exist anywhere, but they are a fair chance of that you can construct that.
Back there, please.
So I thought the Dishaki, the metabolic component, was obviously very useful, since you can plug it directly into experiments. But in the transcriptional case, and expanding on the previous question, even if there weren't too many error bars on the parameter values, even if parts in some ideal world years from now are well characterized, when we actually build the networks and you follow single cell traces, for example, they look nothing like the idealized traces that you fit for. So how would you expand your scoring function to take into account sources of noise and the actual, even extrinsic noise?
Yeah.
And does that add to the because that would be really useful as a
and just interesting questions. I think that we have worked on that, because that's true. To ask to genetic system to behave with a given amplitude, sometimes you only want oscillation, and then come on, you don't care if the amplitude is twice, okay? And then so we are we have in our new version a coarse grain version where just you are higher certain level, so it's just a coarse grain, so it's a more qualitative behavior, because just you need that it makes something that you can convince the people that it oscillates. That's enough. So then you have to have a scoring function according to the audience you have. So that is another thing that is very interesting. Okay, we had to work on that. We had one way to implement robustness. So and we are running some simulations on that. And our robustness we are implementing is on the uncertainty of the parameters. There are other type of robustness, and that still we didn't check. But yeah,
What's the biggest networks you've evolved? I mean, oscillators and latches are fairly straightforward to evolve, but what about much more complex functionality? Have you tried anything more complicated?
Yeah. Okay. We have evolved like circuits 20 genes
And what did they do?
Okay, they do in a complicated. Okay, for instance, our JK latches, for instance, that is not trivial because you have. And I didn't go into the details, but we use transfer functions. So we don't use to fit really the score, as I said. What we do is we fit steady states when you have this truth table. Otherwise, you may have the funny things we found out that you may say, now we want the transcription to go down. And then the system goes down before. And then before you give the signal to go down. Then it just violates causality. Okay? But in our system based on Monte Carlo, Prova is extremely fast. So other people doing in evolution using a population, 2 seconds, it's infinitely much slower.
Right, thanks again, all the speakers.